Okay, perfect. Right. So, hello everyone, and welcome back to the seventh Beyond the Edge seminar after our summer break. Today we're delighted to have Professor Heather Harrington with us. Professor Harrington is a professor of mathematics and Royal Society University Research Fellow at the University of Oxford and a director at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, where she also leads the Center for Systems Biology. So today her talk is entitled Topological Data Analysis for Multiscale Biology. And with that, I'll just hand over to you, Professor Harrington. So thank you very much.
Great. Thank you so much for the invitation to be here and tell you a bit about our work for the Beyond the Edge community. Um, yeah, okay. I've just looked. I have a typo on my first slide. So topological data analysis for multiscale biology with one P in topology. Um, right. So I wanted to start my talk um, with some motivation for why we're interested in multiscale biology and where higher order networks and topological data analysis can be helpful. So um, I've I'm now in a biology institute. So oh, I'm now in a biology institute, and so right next door we have experimental labs where they are generating and exploring complex systems of living systems. And so here is a video, and I could just watch this movie all day long. It's showing how single cells are dividing and developing into a full organism. So this is the zebrafish embryo. Um, and many of my colleagues that are physicists are developing more physics-based approaches to understand self-organization and mechanical reshaping and deformations and restructuring. Um, and I guess where mathematics comes in is to complement these ideas. And so if we look at each of these points in three space, right, we can think of this as a point cloud that's even evolving over time. So it's a dynamic metric space. And we may be interested in, for example, taking one embryo and another embryo and comparing them. And this is like the big, I don't know, vision or program is to develop mathematical models and methods to be able to study this. So, right, so this is kind of the tagline of what we're trying to do is developing mathematics to study the complexity of living systems. And just to tell you a little bit, because we are not too far away, this is the advertisement in some way, is that we've been hiring mathematicians. So we now have three group leaders, one more algebraic geometrics, really focusing on geometry, another one more on the algebraic statistics and phylogenetics, and then the third group leader, Daniela, who just started with us in Dresden, is trained as an algebraic topologist and mostly worked in neuroscience. And so we have a PhD call that's right now open for our IMPRS, or International Max Planck School, and a postdoc call which will go live in a few weeks. And then we have these wonderful ALBA postdocs and a visiting faculty program. So the ALBA postdoctoral fellowships are independent fellowships that allow theoreticians to work in what you see here. This is the building I'm based in, the Center for Systems Biology, Dresden. And right next door is our experimental building at MPI-CBG. And so these ALBA postdoctoral fellowships enable theoreticians to work very closely with other theoreticians as well as with experimentalists. And so right now we launched the visiting faculty program, which we're going to have another advertisement in November. And that's something where right now we have a few visitors that are professors, and it's a wonderful way to exchange knowledge and develop collaborations. So, okay, I think that's my one slide about what's happening here in Dresden, and I have one more slide, which is again a German-based. We're going to have a German-based in Leipzig conference algebraic topology methods, computation, and science, which I thought that this community would also be interested in. And very tragically, our colleague, Shayan Mukherjee, who's worked immensely in topology, statistics, and biology, he passed away last year. So we have a special day that's dedicated to him, and so many of the speakers have connections as well. So I wanted to advertise this. It will be in January, and since it's all local, I wanted to share. So hopefully we can interact in person. And yeah, so hopefully I'll be down in Munich sometime soon. But those are my two advertisements and brief motivation for what we're doing. Okay, so today what I wanted to tell you about, right? This is our tagline of how does complexity arise from living systems, and we really think about this at the molecular scale and building up from molecular interactions to cells and cells forming to tissue organization. And as we cross these different scales, rather than focusing on one scale alone, which one often does in modeling, we really want to bridge these scales. So I'll go through a series of biological examples today where topology, higher order networks, can help study different biological scales and crossing them. So I think it's in the order, but I might be wrong. I think I first start with, I'm going to introduce what is topological data analysis. It's a very quick introduction because I recognize that many people here already know the basics of persistent homology. And so then I'll give an example of where we can use that to study molecules. Then another example where how do we look at hypergraphs with topological data analysis to study worm movement. And then we're going to look at, I think we'll go through cellular environments and spatial transcriptomics. And then a couple slides just bringing it all back together to the motivation at the beginning of spatial temporal biology. Great. So that's kind of what I'm going to talk about today. So what is topological data analysis? So if we suppose we have some points in R2, so we're given some data set, which here are just points in the plane, and we can pick the Euclidean distance. Normally, if we do some type of clustering, we would maybe specify the number of clusters. So we might look at this data and with some analysis say we have 4 clusters. And the idea with TDA is that we want to go beyond picking the number of clusters. We don't want to choose a threshold, but we also miss these higher order features that we would have, for example, with this, let me just see so I can show you. Here we miss this ring, this annulus structure, right? And so the idea with TDA is, which was introduced, some decades ago. It's been building and now very well established. And still growing in many ways, but perhaps the most prominent tool in the topological data analysis toolkit is persistent homology. And so the idea with persistent homology is we look at all these, instead of picking a threshold, we look across all parameter values. And then we can get, the input is this data, this point cloud. We approximate the shape of it by building a nested sequence of spaces on this, basically like thickening the points. And then what we get out is this multi-scale shape descriptor. And it's been applied to many different phenomena: single cell genomics, immune cell organization, chemical structures, spatial networks. So here, if we thicken each point, and as we thicken the points, we then would draw an edge, a simplex of different dimension, depending on the number. So for example, if it's just 2 points that are intersecting for a given radius, then we draw an edge. If there's 3, then here we build a simplicial complex. So we have a triangle, and you look at these higher order analogs, and you continue to study the connectivity of the data across these values, this parameter value, for example, radius, so r. And what we see is as we increase and thicken these points, the simplicial complex, it changes the number of connected components, like the number of clusters. It changes we didn't have any loop, so this one-dimensional feature, and now we do, and then we continue, and ultimately everything is filled in, so now we're left with just kind of one giant component. Okay, and so when you apply homology to this filtration, and we do that as we go along this scale parameter, we can see how the homology is evolving, how the shape is changing with this scale parameter. So as we increase, so along the x-axis, as we increase the radius, the scale parameter, we see that there were 20 points, and now at this value, I don't know, there's like 8, and then we continue, and now we have 3 separate connected components, and we have some small features that are loops in H1. And then finally, just before this scale parameter of 1, we see this 1. Can you see my cursor here? Yeah? Oh, okay, great. That makes it much easier. So then you see this one component, this one loop that's appeared, and it lives for a wide range of values of the scale. And then ultimately it disappears at about radius 1.5, and we see that there's an infinite feature that lives, so one connected component. And this is a barcode. So it's a multi-set of intervals where the leftmost component, the birth of this interval, is where a feature is born, and this is where the feature dies. Okay. So this is the persistent homology pipeline. There's really excellent reviews that were first Carlson, Greist, Edelsbrunner, there are many textbooks now, but we start with our data, we build a filtered simplicial complex, we compute homology in different degrees, and then the output is a barcode. And so one can actually look at this in completely algebraically in studying persistence modules of this filtered space, because it has a very nice structure, and that allows us to get this decomposition of finite and infinite features, and so the barcode completely characterizes these isomorphism classes of the module. So algebraically, it's very nice, and then we have theorems where if we change the data just a little bit, the input, then the output that we get, our barcode, or what is the persistence landscape that I'll introduce in just a minute, is robust, it's stable to perturbations. And then we want to interpret it, and I'll give various examples as we're building this. So one vectorization, once we have our barcode, is to build an explicit feature map. There are different persistence images, which was introduced by Henry Adams et al. Is extremely good, and we use that for many different vectorizations. Today, I'm going to talk about, and this is where you have this grid over your diagram, and you convert the points into a weighted sum of functions, like Gaussian functions, centered at each of these points, so you get what looks like an image, and then you can use it for traditional image analysis and machine learning. So in 2015, Peter Bubenik, and then in 2016 with Pavel Dlako, they introduced the persistence landscapes, and this is a functional invariant of the barcode. So you convert your birth and death, your left and right endpoints of your birth-death pairs, into a sequence of functions or landscape layers. So each layer is a piecewise linear function, and it traces the outline of the diagram's points. So at each lambda k, So this is like the first landscape, the second, the third landscape. At each lambda k, it records the persistence of the kth most persistent feature at each scale. So this is the most persistent feature. This is the second. This is the third, for example. So you have that at each scale. What's the first, second, third, um, at each value of your scale. And so an advantage of a persistence landscape is that this is mapped into um, a Hilbert structure. And so then you can take averages and perform statistics, which is not so straightforward with a multi-set of intervals. But it remains all the useful topological information while being able to then combine into more statistical processes, statistical analyses. So this is a persistence landscape. So now I want to give you an example. So if we think of proteins, so this was an undergraduate project where with the PDB, so the protein database, you have the carbon backbone. These are vertices or points in three space. So you can interpret this as a point cloud. Then we built with just the standard Vittorius Rips. This is pairwise distance, this filtration. And then we computed persistent homology. So instead of the barcode, you can have an analogous, the birth is the x coordinate, and then the y is the death. So you look at everything above the diagonal. And this any points away, just like instead of a long bar is persistence, points far from the diagonal are stronger topological features in your data. So we started with the proteins and we looked at the simplest non-trivial, not the trefoil proteins, computed persistent diagrams, and then we can compute the landscapes so we could take averages. But we also are able to pull out the cycle representatives in the data. And so we used Irene, which is a way to compute bases for this, for the algebra underlying it for this chain complex before you get the persistence diagram. And so each point, right, in the persistence diagram corresponds to a feature, right? So this is corresponds to a feature. And we are able to pull out the cycle representatives. It's not unique. These are not canonical. These are not optimal. So you can have many different cycle representatives, potential cycle representatives for the same feature, right? But you do get one for each feature. And so then we can go back and interpret even more. So we did this with the local geometry here. And what we found is that for a family of very similar, so homologous, very similar protein sequences, that we were able to distinguish between the knotted and the unknotted protein just by looking at the average landscape. And we see that the knotted has in the second landscape slightly different than the second landscape of the unknotted. And it perfectly, by using the cycle representatives, it pulled out that the difference is by this crossing change in the knotted and unknotted proteins. So I think this is a really nice demonstration of landscapes identifying differences in what would be really more a geometric rather than a topological difference. And then the distinguishing part is that we have a change in the crossing of knotted and unknotted proteins. Okay, so we did this analysis, and then the next question was, well, why are we looking only at kind of the most persistent features and looking at the cycle representatives with the most persistent features? So now, let's suppose we have some data. We started motivated by the protein curve, but let's start with some other data. We can look at the most persistent, the second, the third, and then pull out what these generators are. And for each, this is a subset of the points in our original data. For each point in the diagram, we then compute a generator, cycle representative, which is a set of vertices, set of nodes in our data. And then with this set of nodes, we have a persistence. So what we did was take this set of points and map that and say this defines one hyperedge in what we then call the persistent homology hypergraph. And the second most persistent and the third most persistent. And for each of these there's a weight, and the weight associated to the hyperedge is the persistence. So what this, how long this bar is. And then again, we did that for the purple points, this subset, and then for the green points again. So then what we, from our point cloud and computing the pH generators, these cycle representatives, we have a set different subsets of our data that define different hyper edges with the persistence weight. So we now do that for all of the different points in our persistence diagram, and we're left with a weighted hypergraph. So this is giving you a collection. The pH hypergraph is really describing which points are most persistent in some way. For us, it's looking at cycles, so we could weight it by the persistence in our pH persistence diagram. So with this hypergraph, then we are really incorporating more of the complexity and the importance of these points and their role in these different topological features. And then once we had our pH hypergraph, then we can use different techniques that we know and love in network science. So we measured centrality and communities. So I'm going to give an example of how we've looked at this in genome organization. So going, instead of proteins, we have chromatin, and chromatin is also, normally you think of the DNA just as a strand, but of course it lives in 3 dimensions. And so you might have parts of the genome that actually kind of curve around and will touch each other and interact. And so looking at genome organization, we have a ground truth, which is a pairwise distance between all the points in genomic regions. And the standard approach is what they called spatial domains. They look at very local interactions. But then when we did this pH hypergraph, what we found with a more TDA approach is we find also communities of this hypergraph. We find communities that are larger, so showing more interactions rather than only very small local ones. So we give more of a multi-scale, and we applied this to a data set of where each node in our chromatin is something between 30 and 60 kilobase pairs, and this is on chromosome number 21. Again, so we're looking at coordinates of the backbone of the curve. And we use this data, it was published in Cell. So I think this is a very convincing, cool idea. And then the second thing we looked at was node centrality, right? Where instead of looking at membership and what communities are in this hypergraph, we now are looking at ranking the vertices in our hypergraph based on importance, and we used a spectral centrality measure to determine which nodes were the most important, in at least one hyperedge. And we applied that then to movement of worms. So we looked at a nematode moving in some agar plate, and as it was moving, we then took the coordinates. So you think of this as a spatial curve again. We took each of these points, we ran it through this pipeline, computing persistence, computing the pH hypergraph, computing the centrality of that, and actually the centrality values very nicely split up, whether it was intensity of a local search, whether it was relocation or a looping behavior. So I think this was nice to be able to then interpret more of what the cycle representatives are in the information from the persistence diagram rather than a bunch of points where you, or a bunch of intervals and what does it mean, more in terms of the interpretation. Okay. So now I'm changing gears completely. So before I was just talking about curves in space. Now I'm going to add a little bit more complexity, which is, biology is extremely complicated. Disease, this is a disease. This is a schematic of cancer, and disease is even more complicated than biology. And this is an ecosystem of cells that surround a tumor, and you have many different types of cells. It's a complex system. The immune cells are interacting. Sometimes they're trying to fight the tumor cells, but sometimes the tumor cells actually kind of convert immune cells to help them. It's a huge field, mathematical oncology, which I'm not going to be able to be able to explain. But the aim is to be able to use this data if we have some spatial information about the way that these cells are organized and where they're located, if we can give some insight into their disease stage or severity in some way, and that may inform appropriate treatment strategies. For example, if the immune cells are really fighting the tumor, then that would help if immune cells, you give some type of immunotherapy. Whereas if the immune cells have been converted and now they help the tumor, well, then maybe they won't use these immunotherapies in the same way. This is beyond my expertise, but I'm just giving you some examples. So based on synthetic data that was developed through a mathematical model with Josh Bull and Helen Byrne, you have cancer cells, and then you have multiple different cell types. You have blood cells and immune cells, and you want to look at the interactions between these cells. So there are different complexes beyond the standard Vittora's RIPS. So there's one called the Dowker complex, or the multi-species witness complex, which Bernadette led. So Iris led more of the Dowker persistence, and Bernadette led this multi-species witness. We get a vectorization, and then we give some biological insight. So I want to just highlight a little bit about an example of how we've looked at this. So as I mentioned, whether you have immune cells, these macrophages that become pro-tumor. So these are or anti-tumor. So M1 are kind of anti-tumor, and M2, the pink ones, are pro-tumor. And we want to predict whether these macrophages become M1 or M2. Or the second problem we worked on is whether the tumor cells, these blue cells, they form a perivascular niche. This is tumor cells around blood vessels. So we looked at both of these questions. And the way that we did it was we looked at building something called Dowker persistence. So if you're given a distance matrix of two different point clouds, so types of point clouds, you can extend standard filtered simplicial complex to what's called a Dowker complex. And there's something called Dowker duality, and there's a proof of Dowker's theorem that allows you to explicitly find these homology classes supported on either your first point cloud or your second point cloud. And these are actually dual to each other. So what it allows you to do, Dowker persistence and the Dowker barcode, allows you to summarize these different topological features that are either common or shared in both collections. And then we could answer these types of questions. So we describe this as kind of relational topological data analysis for these tumor immune interactions. And what you get is, as I said, two different point clouds. So here are macrophages and these are tumors. And so this is a setting where you don't have this infiltration, and this is a setting where you do have infiltration. And we want to be able to quantify and then give some description and interpretation. So Each of these is called a Dowker persistence diagram, and you have six of them. So you can see here that we have the um, right. So let me explain the data first. We have the um, tumor, then we have the macrophages, and then we have the vessels, the blood vessels, which are these crosses, right? And then for each of these, we have three diagrams, and we can see that with six diagrams in each row, the large birth parameters here in the zeroth persistence diagram, so the circles, the circle here is the zeroth persistence diagram, so these are connected components, and the cross is the first Dowker persistence diagram. So this large birth in the A in this in this in this diagram is indicating that the macrophages and tumor cells, so macrophages and tumor cells, are far from each other. So we can see the tumor, we can see the macrophages, and that they're far from each other. And then we can see that all of these small births here indicate that the macrophages and the blood vessels here are co-localized. So they're all very close because basically the features appear and they disappear very quickly because they're very close. So this is giving us information about the relationships between the different points. Here in the persistence diagram between T and V, it's again similar between the persistence diagram with M and V, indicating we have some relationship between macrophages and tumors, and it's very similar to the relationship between the blood vessels and the tumors. So you can see the blood vessels and the macrophages have a very similar spatial relationship to the tumors. Yeah. Okay. So this gives us some way to be able to distinguish and to describe that clearly looking here, we have a very different behavior because everything seems to be, you can see that the macrophages, I mean, this is the visual, right? That the macrophages, did I talk about that you have this common loop? I think I skipped this, right? Yeah, I skipped this. So you have macrophages and the blood vessels, they have this common loop, which is our one-dimensional feature here. And then if we look, we don't have a shared loop, for example, with macrophages and vessels here. Okay. So this relational TDA, I think, has a lot of potential, and this is something we're actively exploring. I've showed you in a synthetic example, but now also we've applied this, and this is with led by Iris Yoon, but with Eric Sahai, Helen Byrne, and Charles Swanton. So this is a collaboration with London-based researchers. So I showed so far standard persistent homology with one feature, with one type of data, and then Dowker persistent homology with two features. So we can see the zero-dimensional Dowker persistence is giving us like exclusion regions where one set of points and another set of points, here it's leukocytes, the immune cells, and the cancer are in different places. Whereas the one-dimensional features is showing where you have these shared loops in the data. So this is giving us different information that we can then interpret in real experimental data. Okay, so I think I'm about halfway through, which is perfect. It's 11:30. So now I want to go, I've kind of talked about the molecular level, then I've gone to cell level, and now we want to bridge these scales. And I'm very excited to tell you a bit about some spatial transcriptomics, which is a relatively new technology. It's been around for about 5 years. And with this technology, we're measuring information of the gene expression, the transcriptome. So you have the DNA, and then you have something called transcription, which then you get the transcripts out of the RNA molecules. And then these RNA molecules are then translated into protein. And the protein is a little bit more tricky to measure the whole uh, transcriptome, the whole proteome. So we often get data of the transcriptome, even though protein kind of represents function. So you have this somewhat proxy for cell function by measuring the full, 20,000, you get about 20,000 different genes that are measured in transcripts. Okay, and so there's different transcriptomics paradigms. So um, I'm summarizing what took us like more than a year to learn biology-wise, but I'm going to show it to you in by pictures, which I think I wish this would have we would have learned this way. So we have these different cells, the molecular level, and they have genes that are being expressed. And there's different technologies. So when I was a PhD student, uh, this was all expressed with bulk. So this is saying, okay, we have all this tissue from a biopsy, for example, and you chuck it all together and you blend it all up, and then you look at, okay, what does the gene expression look like as a whole? What was in all of the cells? And then came around single cell transcriptomics, and that's been, I mean, this is kind of standard in um, most molecular and cell biology uh, analyses. This is where you measure, you have your biopsy or whatever, and then you run the cells through, and as the cells go through, they spit out this 20,000 dimensional vector, which is the gene expression. So you know where each cell, the gene expression of that cell. And then often the cells are clustered into cell types based on gene expression. Now, spatial transcriptomics is a completely new paradigm, last 5 years, and this is where you can measure the gene expression, this 20,000 dimensional vector, and you record the spatial localization, like the X, Y, Z coordinates, but without knowing which cell it came from. And so, kind of one of the big questions in the field is how do you measure the information that you get from single cell data, where you know what the gene expression is of each cell, with the spatial information? And this is really difficult to do if you can't see cell boundaries in your tissue. So right, so this is spatial. I think that this is my introduction to transcriptomics in 2 minutes with pictures. Okay, but then you kind of get an idea of these different paradigms. And the way that we looked at this problem is trying to combine kind of with this filtration, this nested sequence. So you continue to aggregate information you have spatially until you have enough confidence to be able to say, okay, this gene expression profile or pattern looks like this cell type. Right. So here's the cell type classification problem, which is that we have some tissue. Let's suppose we have ground truth, which never happens in biology. So we do some type of synthetic data, right? And we have this in situ measurement of RNA expression. And instead of, one per cell, we have many per cell. So this is subcellular without boundaries. And the idea is that we want to distinguish the different cell types from the spatial transcriptomic information so we can classify them. Okay, so we want to classify these spots. And the challenge is that normally we fix the bins and then you lose any of the rare cell types, like I showed you earlier. And that's really important because in many diseases, the rare cell types actually are extremely important, like these immune cells. So we misalign. You can either misalign the grid, right? So you lose the sparse cells, or you need different bin sizes for different cell types and sizes. So we came up with this topological automatic cell type detection where we classify each spot independently and we build a neighborhood graph in some way, like a filtration around it, and aggregate the transcripts at each spot. And we do that as we increase the resolution, increase the scale of this until we have a confident enough value. And then we predict per spot what cell type it is. And we actually applied this to the work from the original StereoSeq data. And we're able to identify immune cells, which the original data set was unable to do. So just by pictures, we have for each spot, we aggregate these expression vectors. We predict cell type with a confidence region. And we do that for every single spot. And then we apply this to a synthetic example. So we just randomly generated some cell centers, create a Voronoi diagram to give cell boundaries, randomly assign some synthetic cell types with a, a realistic model to simulate subcell grids. And then we applied it to the synthetic data where we have different cell types represented by different colors. This was the state of the art at the time, so it's called RCT-D, and they looked at fixed bins, and we used TopAct, which is really bin one, right? So we're doing it at every single cell, every single spot, and if you compare them, we get a lot more of the features which are lost for kind of these rare points, right? So in our synthetic data, we have no spots that are picked out. Instead it's only this here, and we're able to pick out some of these more subtle cell types. So we apply this to the synthetic data set. And one of the advantages is that we don't have to bin. So we have subcellular, really fine resolution. We don't have to bin it into multicellular to decompose it. We go directly from subcellular to single cell interpretation. Okay, so the motivation, and that's how we came across this problem, is that there were some nephrologists and they were looking at this problem called lupus nephritis. I'm not a nephrologist, but I will tell you a little bit about the kidney, which is super cool. I think it's super cool in so many different ways. So within the kidney, which is the filtering system, they have these little regions or substructures in the kidney called glomerulus. And in the glomerulus, with lupus, which is an immune disease, there are immune cells. And the question that we had was, given some spatial transcriptomics of mice kidney, can you determine the cell types? Okay, so right, and with lupus, you have this infiltration. So the immune cells affect these glomerulus regions, the filtering regions in the kidney. So we applied this idea of assigning with some confidence what cell type it is to this human kidney data as well as mouse kidney data. So I'll show you human first. H&E is like a normal biopsy. So this is what a pathologist looks at under the microscope. And then the state of the art, it looks like this. So you lose a lot of the structure, and our approach was able to with this TopAct was able to give a more faithful visualization and quantification for cell types and the location. So when the clinicians saw this, they were really excited that we passed kind of the state of the art. And then they said, okay, well, how does it work with disease with mice? So we had the spatial transcriptomics. We have 4 controls and 6 that have this lupus-like treatment. And the aim was to identify and describe the immune cells. So we applied TopAct on both of these kidneys, the 2 conditions. We've pulled out the immune cells, which is great because it was not possible with the existing approach. And then what was the nephrologist we were working with was like, oh, that's really interesting because it looks like you're finding more immune cells near these glomeruli. And we know that there's some type of infiltration of immune cells in these glomerulus regions, these glomeruli. So they said, okay, is this statistically significant? It is. So then we can give some statistical measure that this topological inspired approach can distinguish in a statistically significant way the immune cells. Okay, that's great. Then the next question was like, well, can we say anything else? Okay, and this is where, so far, vanilla persistence, then we talked about Dowker persistence, but now if we have some type of outliers, so there's a bit of noise, which always happens, we want to go beyond standard 1 parameter or this relational TDA to multi-parameter. And multi-parameter persistence was first introduced by Carlson and Zomorodian in 2009 and 2010. And they proposed extending single parameter persistence, so where you get these barcodes for very persistent features, to to two parameters. So considering density, right? Because we are losing, once you have outliers, so there's points here, you lose this strong topological signal, right? And so if you're interested in getting learning more about this, actually Magnus Botnan and Michael Lesnick wrote a beautiful review of multi-parameter persistence. So it's Botnan and Lesnick. And so here you want to introduce the idea with multi-parameter persistence is multi-filtration. So with two parameter persistence, you have a bifiltration. And the idea is that you have your scale parameter, normal parameter, single, this would be normal persistence, but then you have, you also filter by your density. So if you consider all the points and you forget density, you wouldn't see any features here, right? Because you have noise in the middle. So you'll never see a persistent feature. But by considering the density, codensity is just 1 minus density, you're now filtering by two parameters. And so this is really cool. I'm getting that to why it's cool for a number of reasons. There's mathematically, multi-parameter persistence does not have a barcode. So, and as I said, the barcode is really nice because it gives you the isomorphism classes and of your module, whereas computing, since there's no barcode, there's no decomposition theorem with multi-parameter persistence, there's many different invariants that are proposed. And I guess there isn't consensus yet of what is, many people use what's called the rank invariant. And then building on the rank invariant, there's a analogous landscape, what I introduced for one parameter persistence by Peter Bubenik. Oliver Vipond, who is a PhD student in Oxford, proposed the multi-parameter persistence landscape, again, assigning a scalar value to each choice of parameters. And so this is what a landscape looks like in multi-parameter persistence. And you can see if there's a large loop, then you have a large signal for your radius. If there's a small loop, you have a smaller, you have a signal of this H1 at a smaller value. But if you have a noisy loop, which is the case with many biological systems, there's some noise, you would get nothing, you would have no features, right, in your, in H0, I mean, in single parameter persistence, but you have some signal in H1 here with the multi-parameter persistence landscape. So we applied that to the spatial transcriptomics of the cell spots for both treated and controlled from the immune cells. And then we then can take averages because they're landscapes. So these are very amenable to statistics. So we do this for each patch and we see there's no signal with the control and there is a signal with the treated. We take an average and we predicted that this infiltrating immune cells actually form a ring structure in the glomerulus. And then this was experimentally validated. And then we could look at different immune cell types. And so this is the experimental validation of these immune rings. So I think that's very cool that we were able to use kind of a advanced, or at least state-of-the-art, multi-parameter persistence landscapes to study a relatively new technology, spatial transcriptomics, and actually confirm a biological hypothesis that we made. Okay, so I hope I've convinced you today, or at least here's a couple more ideas of where multi-scale biology and persistence can be useful with different barcodes and different pipelines, whether you're looking at a single neuron here, or you're looking at groups of cells. So these are little mini organs, or you're looking at really dynamic spatial networks, whether can be synthetic. So I gave you examples of synthetic models of experimental data. It's the same thing, synthetic models, spatial networks, and experimental data. And we're trying to combine these in different ways with persistence and parameter inference and model selection so that we can also extend to kind of, for example, voids and what's not so easy to extend with, for example, standard network and standard kind of one parameter persistence approaches. Um, and all of this ties into some machine learning and interpretation. So the final place I want to leave us today is talking about spatial temporal persistent homology. So if we look at this data set that this is from Ian Cousin, who's one of another Max Planck director, and he's looking at a lot of collective dynamics, and this happens in biology and physics, right? We can think of fish or birds swarming. Yeah, there's many different computational and agent-based, so agent-based models and real live tracking. And the challenge here is that we don't have a nested sequence of spaces. There's not a single barcode that satisfies this, right? Every example I showed you is you start with your data and then you build something that includes into the other. So it's like, monotonically increasing. And in 2019, Facundo Mamoy and Woojin Kim introduced what they call an interlevel RIPS trifiltration. So instead of a bifiltration where there's two, there's actually three different parameters to be able to consider scale and time. So basically you think of time and you either look at time and you're going forward or you look at end time and you go back, and then you have a filtration with these two time intervals and you have space. The problem is three parameter persistence is not, there's no way to compute a good basis for a three parameter persistence module and diagram landscape. So this is something that is also in general to get this basis. One way to do this is compute Grosvenor bases. And in general, this is doubly exponential. So this is work that's in progress by colleagues Matthias Bender, Oliver Gavert, and Mike Lesnick, which they've introduced, or they're working on this GBS algorithm. They consider finitely many components of this multi-parameter persistence module. And so you don't have to consider, so by considering just some of these components, it's enough to get your Grosvenor bases. And then they have combined, basically there's two different approaches, what is called F4 or F5 for Grosvenor bases. And so they've been able to combine this in such a way that they're able to, their procedure, they're able to have a finite number of submatrices, that's point one, and also be able to reduce the complexity with this clever choice of some ordering. So then it's only exponential, not doubly exponential. Okay, and so that's active work, which I'm also going to share some active work we're doing, which builds on what they've done, which is computing a landscape, where naively you have to compute all the different ranks. So it's O and to the 2R, where R is the number of parameters. And our contribution is that we can compute landscapes more efficiently, where again, we only have to look at a subset of the Grosvenor basis, and we can then compute it instead of ON to R, ON to the R. So this is, instead of looking at naively looking at all possible ranks of computing the ranks of all possible maps, we can reduce that to the square root of the number of operations of this naive algorithm. So this is all like in progress work, but if we look at a bird swarm by Chad Gusti and Derek Lee, they published this, where the reason I like this data set and I want to show it to you is that the cyclic behavior is not at a fixed time, right? You never see the cycle. If I fix a time snapshot, you never see the loop. It's a dynamic process. So topologically, we can only see it in the dynamics. And then we applied this three parameter trifiltration and then the landscape. So we fix the time interval T2, and this is a slice of that. And we can actually see that you have this feature at later, um, kind of as you're going T0 to T1, you have this feature in later time of what this kind of ring dynamically. Okay, so I think I've spoken for 52 or 53 minutes, something like this. So this is the future, I hope, of spatial temporal TDA. And thank you so much for having me kind of share how biology motivates new with models and data, it motivates new kind of higher order and topological data analysis approaches to then give predictions and test them and then again new insights to learn more about the biology. And today I hopefully convinced you across whatever scale of data that you're studying that TDA can be useful. So with that, I want to thank you for your attention, thank funding and our growing group here at MPI-CBG, and I hope you can, yeah, I obviously welcome questions, but I also hope that many of you consider visiting us. So thank you.
Okay, thank you very much. It was an excellent talk, really insightful. So yeah, are there any questions? Um, I'll just you can unmute yourself if you'd like to ask one. Okay, well then I think I might start actually. I would ask one. You have this Vittoris Rips construction at the beginning, and always you
Hmm.
filter by this epsilon parameter. And
Hmm.
typically this is the Euclidean distance between points, if I'm
Hmm.
not mistaken.
That's right.
But I was wondering, can you instead do this by maybe some sort of interaction strength between two cells? And because I wonder then in such a case, are you guaranteed to have a simplicial complex if, say, for instance, a strength only goes one way, but it doesn't come back, it's not reciprocated? And then maybe can one then still construct such a persistent homology diagram?
So you don't have distances between all the points? Is it
Yeah,
you just need a metric on
just
your.
any metric. Okay, right,
Mhm.
right. Okay. Because is the simplicial complex the most fundamental object that's required in this construction? You
Right, you
can't go
have to build a
any.
simplicial complex before you can do.
Okay,
Yeah, even like when people work with often, if you work with hypergraphs, there are different ways that people then compute persistence of a hypergraph. You then somehow, right, build some simplicial complex.
okay, perfect. And then in this bipartite data, you had the Dowker complexes were essentially what you were using mostly, but then later on you had the multi-parameter. Could you use both in one setting and kind of compare how the two go against each other?
I don't think it's been done yet. I think it's hard with Dowker. I don't think it's been done yet, but it's something to think
Okay,
about.
okay, interesting. All right.
Thanks. Yeah, that's
Yeah,
interesting.
yeah, yeah. Because I know you had it in this tumor versus immune cell interaction. I was just wondering if it could be.
Yeah, that's
Yeah.
right. Yeah, I think that's a really good question. I think that
Right.
there's some math probably to work through.
Okay.
Mhm.
Okay. Any other questions?
Yeah,
Yeah.
maybe I can come up with a question. So, hi, Heather. I'm Dan.
Hi.
So, actually, I had to answer a phone call, and because of that reason, I missed a part. Maybe that's the reason that I couldn't get. But so it's quite interesting in the special transcriptomics data. Then you are basically finding deconvoluting these things and from the single cell expressions. Then afterwards, you are trying to find the alignment of the cells. But in the figures, this is correct, right? So this is what I understood. I mean, so and like I just couldn't understand this in the presentation, like about 40 pages, and
Uh-huh.
you do have this grid shape,
Oh.
and then for each spot you show like a single cell is there and so on. But my understanding, this is like a bulk, right? So as you said, like here, for instance, this.
Mm-hmm.
What does this, for instance, pink, green, and then purple means on the right?
This is like a 20,000 dimensional vector that I've just shown a visualization of. So,
Yeah, probably I missed that. Yeah,
right,
so
right, right. Okay. So, yeah, maybe if so for each spot, for each XY coordinate
yeah,
here, we have a vector. So this is there's different transcriptomic technologies. So some are the whole genome. So this StereoSeq, which is what we're using, is array based rather than image based. So you actually get this 20,000 dimensional vector of
Yeah.
the genes, and you get that, but it's very sparse because it's so small. And you get that for every single spot. So you basically have, you could think of even as a tensor, X, Y, and then 20,000 a vector,
Yes.
20,000 dimensional vector for
It is
each,
aggregated, right? Somehow from different
sorry.
cells.
And we don't have the cell information. Now people take we don't have like the cell image, so we don't see the boundaries here.
True, yes, I
Now,
understand. But this is
yeah.
exactly, I mean, so like the special transcriptomics data, right? For each
Mhm.
spot, I mean, so you do have multiple cells in it, and then you are basically counting, aggregating all the genes as a vector.
It's subcellular. So you don't have all of um, so we have, I think I took out the slide with all the dimensions. Let's see, did I? Yeah. Um, yeah, I took out the slide. I usually have a slide that has all of the um,
Okay, maybe
yes,
I
but
couldn't
yeah.
understand what kind of data you really in this, like what is
Mhm.
really the subcellular and so on. So maybe that's why I couldn't understand. But at the end of the day, so I see this like green one, pink one, and then the purple one, like as cell types, right? If you go a slide further,
Yes.
just a slide further,
Yeah.
40, yeah, this one.
Yeah.
And then now, I mean, so in this each spot, you show one cell type. So how can it be possible? This is not clear to me.
Okay, right. So there's the other information that maybe I can't remember if I said or not is that for each sample, we also have kind of some type of you want to how people define cell types is interesting, but there's some reference in some way of like these gene expression, certain markers correspond to this cell type.
Yeah.
And so then we, from that information and from our aggregation of transcripts here, as we're doing it per spot,
Mm-hmm.
then we can use this as a reference with
Mm-hmm.
enough transcripts to then give some confidence. I forget what it was, like 75 or 80% confidence
Whatever.
of which cell type it is. And the reason we have to aggregate is that I think the diameter of each cell is like 10 micron, but then the diameter of each spatial bin in subcellular transcriptomics is like 200 nanometer or something. So you really need like 400 or something like this is approximately per cell, but then some cells are different sizes. So we have to aggregate enough because we don't have enough transcripts to do it
Sure.
really truly individually only on each transcript.
I mean, okay, I couldn't really get still, but maybe I need to read your paper and drop you an email. Then afterwards, maybe we can chat about this because this is very interesting to find the alignment. But if this is like on the right picture, right matrix is a description of special transcriptomics data presentation, then I cannot really understand how you can assign a cell type to a spot.
Well, I mean, for each spot, it's a vector, right?
Correct.
Yeah.
And
And so
then
then
you do have like multiple cells, right?
for each spot, no. So that's what
Okay.
I was saying. You need almost 400 because it's extremely sparse. So in single cell, not
You
spatial,
say,
you
I mean,
have,
so for each spot, you can only have information of a single cell.
yes. And sometimes there's dropout. So like if you're not right at the nucleus, you almost have no information. But of course, we can't see whether we have the nucleus or not.
Okay, I'm not aware of this data. Okay, all right. So
Uh-huh.
now I understood. So I was confusing with the like a
It's
traditional.
not the aggregation, right? It's not, let me just see if
Okay,
I
now I
can,
understood. I'm
yeah,
so
I think
sorry.
I tried to, let me see if I have
Sure.
it
Now I
here.
understood. I'm so
Yeah,
perfectly. All right,
yeah,
brilliant.
okay.
So thank you. Thank you for
Yeah, good,
clarifying.
good. You're welcome. Yeah, I think I tried to put in this because I find it, yeah. So from subcellular, where you have many spots per cell, instead
Yeah.
of aggregating it, which is what most people do with bin 20, they aggregate it and then they decompose it. We're just trying to aggregate only a few cells to go right to the single cell
I
prediction.
see. I understand, but
Yeah.
I didn't know that there is such a technology that each spot is basically smaller than a cell size.
It's newer. It's newer. So 2020 when it first arrived, it was exactly what you said. The first types were
I see.
multiple cells, but then
Okay.
now the spots are
Cesium,
so
something
much
10x and
smaller.
so on, right?
Yeah.
So,
Yeah, exactly. Yeah.
okay. I'll check out this
Thanks
out.
for your question. Thanks.
Interesting. This is very interesting. Thank you.
Okay, thank you. And are there any other questions? Well, well, if not, then I would like to thank again Professor Heather Harrington for excellent talk, and thank you all for coming to
Thank
the
you.
seminar. Okay,
Thanks
perfect.
so much.
Thank you, Heather.
Nice
Thank
to
you,
see you,
Heather.
Rafaela. Thank you.
You
Thanks
too.
for
Thanks
Bye
again.
bye.
Bye bye.
Bye.